Welcome to Your Brain, a Personal Tour. I'm Steve Saltwick, your tour director. In this seminar, we will tour the machinery that is sitting between the ears of every single person listening, your brain. It's both a marvel and a mystery. The marvels we'll get to in a minute, but the mysteries will not only intrigue you, they'll also give you some very cool snappy patter at cocktail parties. For example, why does holding a cold drink in your hand affect your judgments of the people around you? What do nuns do or not do that help prevent Alzheimer's disease? Just what kind of moral decisions does a 6-month-old make? And they do make them. Why is the fact that your brain has single cells that recognize Jennifer Aniston or Ryan Gosling a sign of an advanced brain? And finally, why don't lions just eat everyone on a safari vehicle? Interested in the answers to these mysteries? Well, okay then, let's get on with the tour. The landscape of our tour is on the following slide. It will be one of the few photographs of the actual brain. It's a bit bloody, so be forewarned, we'll only see it briefly. This is the object of our tour, the human brain. We'll see lots of pictures in this seminar, but very few of them actually of a human brain like this. Why? Well, this organ is more complex than the galaxy we live in. Billions of cells and trillions of connections. There is just no way we could fully understand what is happening on this tour without simplifying our study. And we've got a good friend who can help. The lab rat. Even though her brain is much smaller, we share far more brain structures with a rat than many would care to admit. Many of the points I will make involve rats, but I do so that we can better understand the human brain. Our knowledge base would be much, much weaker without the contributions of this little dude. So if you can, hug a rat during the course of this seminar. I will also note here, rather than repeating on each section, that great care is taken to humanely treat rats or any other animal in experiments that I describe. Full medical support is used. For example, anesthetized surgery, ethics review of experiments, etc. Every effort is made to minimize any pain or suffering during the course of experiments described. We'll also tour the brains of monkeys. Here, a monkey's brain is controlling a robotic arm. More on this in the last seminar. Monkey brains set the stage for human brains, so we can learn a lot about memory and learning from monkeys. And we've learned a lot about brain repair from monkeys. Experiments like the one shown on this slide have progressed medical science substantially in just the last few years. Finally, one housekeeping note. All of the seminars are on YouTube. See the program website for more information. Closed captions are also available. And if you get those annoying web ads, just click them off. Our agenda is structured around 6 seminars. We begin with perception and movement, and we end with brain aging, dysfunction, and repair. I'm sure you're anxious to begin, so let's go. Our seminar today concerns perception and movement. My one-sentence summary of the seminar is, quote, what you see is not what you get, nor how you act. What our eyes see is not what we perceive. How we think about moving is not how we move. How can that be? Well, welcome to the seminar, and let's find out. We begin with a document that's 4,000 years old. This papyrus was written by a doctor in ancient Egypt and records his observations and prognosis on a very wealthy Egyptian. Called the Edward Smith Surgical Papyrus, it contains this hieroglyphic. This hieroglyphic is translated as brain. So this is the first recorded use of the word brain in a written language. The study of the brain has fascinated mankind for over 4,000 years. Much has been learned, but much remains to be learned. When this doctor was examining his patient 4,000 years ago, he had only a primitive understanding of this wondrous organ. A bit of progress has happened in 4,000 years, so let's take a look at what we can do today.
To Roger Frisch, the violin is not just an instrument, it's part of him, an extension of his passion for music, beauty, and excellence. Roger's mastery of the violin has put him on the world stage. Few have reached this level of expertise. But not long ago, Roger noticed something wasn't quite right when he pulled the bow across the strings.
When I would draw my bow, I suddenly had a shake in my bow. Now, for most other professions, this would not be a concern. For being a violinist, where your career depends on the stability of your appendages, this was a great concern.
Roger has what's called essential tremor, a nervous system disorder that causes shaking. Concerned, he went to Mayo Clinic, where he met neurosurgeon Dr. Kendall Lee.
And the amount of tremor he has would be considered pretty mild, but not in the case of a concert violinist.
Dr. Lee and Roger agreed that deep brain stimulation, or DBS, was their best chance at stopping the tremor. It involved placing electrodes into Roger's brain. But there was a challenge to overcome that created a first-ever situation in an operating room. You see, because Roger's tremor was so small, it would be difficult for Dr. Lee and his team to tell if the electrodes they were implanting were in the best possible place to stop the tremor.
And so what we needed a violin in the operating room.
Kevin Bennett heads Mayo Clinic's Division of Engineering. His team worked with Dr. Lee's team to create a violin Roger could play while awake during surgery.
We built and designed a 3-axis accelerometer that we could attach to the bow so that as Mr. Frisch was playing, we could actually see a plot on a computer screen of the movement that really shouldn't be there.
During the procedure, Dr. Lee inserted electrodes into Roger's brain and moved them to the site where the tremor originated. Once there, electrodes fire electrical signals to hopefully stop the tremor. By measuring Roger's movements, the accelerometer told Dr. Lee if the electrodes were in the right spot. There was no guarantee it would work. Thankfully, it did work.
I thought this was a wonderful challenge to see if this technology could help in this sort of a unique situation.
I started playing right away the day I came home from Mayo. I was back playing with my colleagues with the Minnesota Orchestra 3 weeks later. Without the stimulator on, the tremor is quite pronounced.
You can see the difference. With the click of a controller, Roger can turn the electrodes on and off.
And that's as smooth of a bow as I can play right now.
But when it's on, Roger's mastery of the violin is absolutely clear.
I remain very, very thankful every day that I have had the opportunity to have had this surgery. And, I just do not take it for granted.
Back on the world stage to make music, beauty, and achieve excellence.
Brain stimulation is not just for treatment of motor disorders, nor just for elaborate and expensive OR treatments. An exciting new technology called transcranial magnetic stimulation uses magnetic waves to stimulate the brain. This technique can be done in an outpatient's doctor's office. Dramatic results are being reported in the treatment of depression, a terrible affliction most commonly treated by drugs. 30% of patients seem to be resistant to drug treatments, and this new technology is offering them hope. One of the most prestigious journals in medicine, The Lancet, is reporting significant results. I personally have a friend with a son who has benefited from this treatment. We've come a long, long way in 4,000 years. I first became interested in the brain in the early 1970s as part of the biopsychology program in the psychology department at UT Austin. I was privileged to work in the laboratory of Dr. Michael Gabriel, and we published some of our research in Science. I left UT in the late 1970s and took a tour through the high-tech industry. It was great fun, at least for a while. But even more fun was returning to UT and reestablishing contact with Dr. Mike Gabriel and Dr. Michael Damian. They have been of great help with insights and guidance for this seminar. I really want to thank them. I also want to thank Dr. Dennis McFadden for not only hiring me for my first job at UT Austin, but for thorough and constructive reviews of the early drafts of this seminar. This seminar would not have been possible without his help and encouragement. I also acknowledge that he beat me in the mustache contest of 1975. If you're wondering where's Steve Saltwick in this picture, well, here I am with more hair and attitude and a little less weight than the one in front of you today. Now, your brain is unique in that it's encased in bone. It sits atop a spinal cord protected by vertebrae. Nerves exit the vertebrae to innervate not only muscles, but also internal organs of the body. Sexual organs, your digestive system, the liver, organs such as the heart, the lungs, the kidneys, and your stomach. Other nerves pass information back to the brain from sense organs like your eyes and mouth. If we look at your brain once we remove the skull, this is what you'd see. The frontal lobe is at your forehead. The occipital lobe is in the back of your brain. In general, there's not a mapping of function to brain lobe. Yes, visual processing is primarily located in the occipital lobe, and executive functions tend to be in the frontal lobe, but most everything is spread out all over the place. That gives your brain a great deal of resilience to withstand damage and trauma. The size of the lobe is not an indicator of brain cell population. That small bit of your brain at the lower right, the cerebellum, contains 50% of all your brain cells. The cerebellum controls fine-grained pattern movements, for example, tennis swings. So if you ever want to show someone that you can use 50% of your brain, just strum your fingers or swing a tennis racket. That'll show them. Another way to look at your brain is to see how parts of it are connected to one another. This is called a connectome. For example, the brain on the top seems to be connected in a vertical fashion, and the brain on the bottom seems to be connected in a horizontal fashion. Now, these connections have effects on behavior, and that's because the vertically connected brain is a typical male brain, and the horizontally connected brain is a typical female brain. These connection geometries probably explain some fundamental differences between the genders, including motor and social behavior. Further research is going to be fascinating. If we go inside the brain, we find the cells that make up this marvelous organ. We would see billions and billions of specialized cells called neurons. These neurons are electrically excitable and transmit impulses of electricity down long, skinny parts to affect other neurons. When that electrical pulse, the little blue pulse, travels down to the end of the snaky bit, it causes a chemical to be released onto the next neuron. That chemical may or may not cause the next neuron to get excited. So neurons are both electrically excitable as well as chemically excitable. That's two ways we can control neurons, and we'll use that fact in experiments we study later on. That also means neurons can become dysfunctional in two ways, electrically and chemically. Since the neurons use electrical signals to communicate, we can actually record this in living brains and amplify the sound. I'd like to play for you something that we might hear in the laboratory if we were to record from a living brain. There are many different rhythms. Some are quite fast, some are quite slow. Some are silent and burst. Each style has its place in the way your brain operates. Since it's fully enclosed in skull, your brain needs some help to interact with the physical world. That help comes from the peripheral nervous system, a system of nerves that's outside the skull. Everything your brain senses is carried by incoming sensory nerves called afferent nerves. They carry the familiar senses like sight, hearing, and taste. But note, internal sensations from muscles and organs are carried to the brain as well. These sensations can help explain some surprising experiments, which we'll see in the next slide. Behavior is called by efferent neurons leading away from the brain. The vast majority of our time in this seminar will be spent on behavior caused by muscles. We'll not have time to give justice to the autonomic nervous system, the flight or fight system you probably studied in high school biology. But I do want to highlight how the feedback loop of these systems are incorporated into higher order behaviors of learning, emotion, and memory. A strong case can be made that feedback from your internal organs and senses underlie emotional responses and memory. Stated another way, your thoughts are not just abstract symbols and language, but incorporate internal bodily sensations. Some examples might help. If I ask you to evaluate the personality of a stranger while you have a cold drink in your hand, you will tend to find the person colder than if your hand is empty. And yes, if you have hot coffee in your hand, you'll judge them warmer. If you evaluate a job candidate with her resume on a heavy clipboard, you will judge her more serious than if the resume is just a piece of paper. Our inner gut feelings are an essential part of our brain's experience, and that fact will be important as we explain and explore how our brain interacts with the world. One more point on internal organs having an effect on your brain. We will see in a future lecture how microorganisms can travel from your gut through a specific nerve, the vagus nerve, and reach your brain. Parkinson's disease has been transmitted from the gut of a mouse to its brain. The beneficial effects of probiotics may well use this pathway to help protect your brain. This is an area of massive research, and dramatic progress is being made. So never, never forget that your gut has a say in the way that your brain works. Your brain evolved. What we show on the left is some representative brains across a wide variety of species. The human brain is at the top left. A mouse brain is at the bottom right. For those of you that are dog and cat lovers, look in the middle row. The dog people can be amused that the dog brain is slightly bigger than the cat brain. I know the dog people will say that's obvious from the start, and the cat people will say, well, that's a small cat. I'll leave it to the dog and cat people to fight it out. While size does matter, it does not really relate to intelligence. There's some very smart birds whose brains would be down there at the bottom right. What does matter is functional areas, and there is a complication on that point. Brains differ in their organization of functional areas over the course of evolution. To use an example, suppose our common ancestors had red, blue, yellow, and orange functional areas. The family tree on the right shows how those areas might move around in terms of size and placement. The key aspect of this diagram is the appearance of new areas of the brain. They bring new function. These new areas are the essential underpinnings of different intellectual capabilities. What you are seeing is an animation from the Glass Brain project. This is an actual real-time image of electrical impulses in a human brain that's being studied in a laboratory. What is it doing? How does it sense and perceive the world around it? Is what it sees, in other words, what it senses, what you get, in other words, consciously perceive, or is it something different? The answer to this question gives us our first main insight into the way your brain works. Your brain senses the outside world through a series of awesome sense modalities. We learned about several of them in high school, and they're the familiar ones: sight from the eyes, hearing from the ears, etc. Most come to the brain through a relay station called a thalamus, number 2 in the diagram. Smell, however, comes direct. This means you can smell when you sleep. We'll use that fact about smell in some experiments later on. But I want to draw your attention to the somatic senses. They're not as familiar to us. The first somatic sense I want to talk about is called proprioception. This is a sense of what the muscles of the body are doing. The top left-hand part of the slide shows a silhouette of the bicep muscles of the arm. A portion of that muscle is then expanded. The muscle is innervated with some sensors called muscle spindles. Nerves leading out of those muscle spindles give your brain a sense of the state of that muscle. Spindles are not the nerves that make the muscle contract. Those are different and they're not shown. Muscle spindles give your brain the sense of muscle tension. That turns out to be extremely important, not only for our movements, but for our perception of self. Your brain tells your body to move and then gets feedback of the state of the movement from spindles. The brain's interpretation of that feedback is believed to be extremely important in creating the sense of self that we typically assign to consciousness. I find it very, very interesting that such a low-level sense within your muscles might be so important to the conscious construction of self. The somatic sense of proprioception can be used for dramatic effect in areas that might seem surprising. Let's look at one example. Here we'll use the feedback path from facial muscles to reduce implicit racial bias. How is that possible? Well, first, let's look at how we measure bias. Something called an implicit association test is typically used in the laboratory. In this test, the subject presses one of two keys to categorize either words or pictures of faces. The words must be sorted into good or bad categories, and the faces must be sorted on the basis of race. In one condition, the same response key, let's say on the right or on the left, is used for both the good words and the white faces. In a different condition, the same key is used for good words and black faces. The key measurement is the difference in response time between the two conditions. Typically, subjects respond faster to pairing of good words with pictures of faces which are the same race as the subject, and therefore they would be measured as showing implicit bias against the other race. It's important to note that the implicit bias test is more like measuring your blood pressure than measuring your height. All sorts of variables can affect measured results, but the test is generally accepted as a valid measure of implicit bias. So on with the test. Could manipulating a somatic sense actually affect bias? Could it reduce bias? That's the $64,000 question. And to make a long story short, it could. What was the manipulation? Having the subject hold a pencil in their teeth so that they smiled. The act of smiling reduces implicit racial bias. Isn't that cool? So unleash your inner optimist and smile. It will do the world and you good. But humans are really visual animals. Hearing might be the most regal sense, but the sense we typically are most aware of and use is vision. The nerve pathway from the eyes lead to the back of the brain, that's the occipital lobe, very directly. They do crisscross and make one synaptic connection along the way. Most visual processing begins in that back part of the brain. The central part of the eye is very, very dense with sensors, so the central part of your visual field is where you get the most resolution, and that's where you can see the most clearly. But there's also an early warning path in the visual system. Just after the nerves exit the eye, a special path connects to the threat detection areas of your brain. The resolution of this path is not very accurate. It goes on general shapes. For example, snakes, probably the first predator of mammals, and fearful eyes as detected by large relative areas of whites. This early warning path was very helpful in reacting to saber-toothed tigers in the past, and very involved in implicit bias in the present. More on that a bit later. Using computers, we can map where the central part of our eye, called the fovea, is being focused. In this diagram, the little red dot basically tracks where the fovea is focused. That red dot is where your brain is getting the majority of its information about the visual field. Let's play a short video and watch where the fovea tracks. So what did the fovea focus on? To me, it was almost exclusively the faces of the two people. You didn't see the red dot survey the room. You didn't see the red dot look at the clothes in the closet. It almost exclusively focused on faces. The other part of your eyes did take in the visual field, but with far less detail. Your brain builds up a scene over time, and some parts of that scene are getting virtually all the visual attention compared to the other parts of the scene. That's an interesting way to build a picture of the world. After the eye has processed a visual scene and nerve impulse have gone to the back of your brain, how do the next stages of visual processing occur? Well, the first level of visual processing is the detection of elementary objects in very precise locations at very precise orientations. Here we see a monkey in an apparatus where we can study his visual cortex in the back of his brain. In front of the monkey is a screen. We can project lighted bars or other objects on this screen, and we study the response of neurons when we present these visual stimuli. On the right is a figure of what a representative brain cell, a neuron, does in response to a particular bar of light. This one is preferentially tuned to a particular stimulus, a rectangle oriented at about 40 to 45 degrees from the vertical. When a bar of light passes the visual field of this cell, it responds with a burst of activity. That's a very robust sound. If the stimulus is vertical, you get a slightly less response. Tilt it the other way, and you get no response at all. This is an excellent description of how first-order visual processing occurs. Neurons in your visual cortex are tuned to very precise stimuli at very precise places and very precise orientation. What happens after the brain processes this level of detail? Well, various other areas in the back of your brain, the primary visual cortex, analyze the visual field for further basic information. Color. What are the areas of color? Motion. Are there parts of the visual field moving, and if so, how are they moving in relation to one another? Detecting that those set of spots moving behind the little bush next to you could be a saber-toothed tiger, well, that's a good thing for your brain to be aware of. The form of the image is analyzed. Does it correspond to objects that we might recognize from memory? Finally, you have two eyes. We are binocular. We can detect depth of objects, so a 3D image of the object is built up in parts of the brain that give us a sense of solid form. Okay, now what happens next? The next stage is breaking down the visual image into fundamental building blocks. Examples of those building blocks are given in the left-hand part of the slide. They're called geons. They're canonical visual objects. You can think of them as Lego blocks of fundamental shapes. By taking these fundamental shapes and merging them together, the brain is able to build representations of more complex objects. For example, if your brain wants to build an object of a paint or water bucket, it takes the shape of a curved handle, number 5, and a cylinder, number 3, and it puts them together to form the object of a bucket. There's good evidence that fundamental analysis by your visual cortex merges together geons in various ways to form object representations. This is a core principle of your brain. Objects are analyzed all at the same time into simple canonical forms. Then those canonical forms are used to build up more complex objects by additional layers in your brain. Further evidence that your brain works in this way comes from the field of artificial intelligence. That is the computer simulation of how your brain works. On the left of this slide, we see how a 5 layers computer program, programmed to emulate your visual system, processes objects. Each layer learns from the layer below. Very simple shapes, mostly edges, are processed in layer 1. By the time you get to layer 5, which is really not that many layers, the computer program is decoding some pretty complex shapes: dogs, eyes, wheels. Your brain has many more layers to process objects. So how complex can an object be? Well, you might be surprised.
Those biomarkers and our capacity to measure them leads us naturally to Jennifer Aniston.
If we could have the next video. So we have very rarely the occasion to measure the response of the brain at the level of individual nerve cells, because for obvious reasons you can't access the nerve cells in a human brain. We can only do it by and large in experimental animals. But sometimes, particularly during neurosurgery, you can actually listen to individual neurons. Now, inside the brain, it's silent, right? There's no sound. But if you put in a microelectrode and you can listen to the individual nerve cells chat to each other, you can hear, in fact, I think we have a recording, they have the sort of staccato sound. If you take the electrical signal and amplify it and put it on a sound speaker, you can actually hear them talk to each other. Each one tick is a little action potential, it's a little pulse. So this is actually a recording from a surgical measurement where the neurosurgeon put a microelectrode into the brain of a patient, and this nerve cell responded very selectively only to pictures of Jennifer Aniston.
I think it's fair to say Team Angelina.
It is true. It is true. It was because why Jennifer Aniston? It's not that we're born with Jennifer Aniston neurons, at least hopefully. But we're born with a disposition to learn things So, you know, your mother, your parents, the president, famous actors and actresses. So here, by mere chance, we happen to come upon a neuron that selectively encoded the presence of Jennifer Aniston. And sometimes you find cells that even respond to the name Jennifer Aniston or to the name of the famous person. It could be your friend, could be your mom, could be a famous actress.
I'll note that during the surgery, this particular cell responded to pictures of Jennifer Aniston, and not just in full frontal face, but also the side views like you might get on a police lineup, to line drawings of Jennifer Aniston, even the name Jennifer Aniston written out in block letters. Similar recordings have been made in different patients in reaction to pictures of Homer Simpson and other comic characters. We'll see some examples in later seminars. We can show that the brain learns to group objects as a function of experience by studying the perception of newborns. Thanks to the work of one of Ollie's professors, Les Cohen, we can see an example of that learning on this slide. At 6 weeks of age, newborns have no conception of angles. They only react to the orientation of lines. So these two figures are equated as equal since this line and this line are the same. However, by 14 weeks of age, babies have grouped line segments into objects, where the interior angle is significant. At this age, these two figures are judged the same, since this angle and this angle are the same. Take a look at this diagram. What do you see? There's two ways to see this. One way is a set of blue lines and Pac-Man-like circles sitting on a flat plane for no apparent good reason. The other way to look at this object is a solid white triangle suspended above a blue triangle in 3 circles. That's the way most humans would describe what this is. Not an arbitrary collection of line segments and funny-looking circles, but familiar objects in space. What you have just done is make a conscious, perceptual decision. Your brain decided the spatial arrangements of the objects. It analyzed the geons present, and it basically decided they represented forms of triangles and circles arranged in space. Just as we talked about in the preceding slides, it analyzed this for color, it analyzed it for form, it analyzed it for depth. The reason this figure is important is that most research now suggests that when your brain makes that kind of perceptual decision, that is when these objects really enter into consciousness. We're really not aware of how the brain is doing fundamental analysis of the visual field, the line segments, the geons, etc. That's not conscious in our experience. But when we decide on a perceptual framework, when we have built the geons into objects positioned in space, that's when these objects enter into consciousness. Research from May of 2018 has actually suggested a mechanism of how this works. On the right side of this slide, we see object analysis in the back part of the brain. But the object has not entered into consciousness. The analysis of the objects only become conscious when the front part of the brain ignites and there is processing both in the front and back of the brain. One way to think about this is when the front part of the brain ignites, the objects are available in a global workspace for the brain. All areas of the brain can access the representation of the object. Without the frontal ignition, representation of the object is only available to the local area, visual processing. We'll see excellent examples of this soon. I'd like to summarize this section of the seminar using a well-known celebrity, Homer Simpson. We'll use him throughout the seminar to help reinforce the main point of each section. So here we have an X-ray of Homer Simpson. You can see that his brain is slightly smaller than the brains we've been discussing, but that's because his brain can only hold one or two important points at any one time. Homer's summary statement is that your brain senses stuff, line segments, geons, what have you, but you perceive objects. This is the fundamental property of sensory perception. What you see is not necessarily what you get, because you only get objects. Objects can be formed from fundamental shapes. Objects, most importantly, can be formed from other objects. Objects enter consciousness, and that's when you're really aware of these things, when the various sensory modalities have been analyzed to the level of an object and the front part of the brain has been ignited to form a perceptual whole. Objects in space and time. That is the main message of the first half of our seminar. Now let's turn our attention to the other end of the spectrum, movement. We interact with the world around us by moving muscles. We change the position of our limbs, we turn our head and focus our eyes. So in the last part of the seminar, we're going to look at how does your brain do that? And more importantly, we're going to see if there's something that is equivalent to objects as we discovered on the sensory side of the equation. We saw how initial sensory input was analyzed at a very low level and then abstracted into an object. In fact, most objects were extracted into objects of objects. Well, is there something like that goes on in the movement system? Let's find out. Our first hint that there might be something like movement objects was discovered in an experiment from 1977. An enterprise young psychologist asked several subjects to write the same sentence with a variety of appendages. This particular sentence is a palindrome, so it says the same thing whether you spell it from left to right or right to left. It's clear from looking at this picture that there seems to be some kind of template that represents cursive writing. The template is expressed in a variety of muscle movements. The right hand with the wrist restrained, the left hand, the teeth, and even the foot. Clearly, fine-grained muscle movements do vary across the samples. But I look at this and say there's some very interesting evidence that points to a particular movement template. That template is then expressed by different movements of different muscles. But again, the movements are expressing the same object, the sentence. In the search for movement templates, there's always been a controversy. Does the purported template reflect the actual goal of the movement itself, or is it really just the muscle commands in some fashion? We need to find neurons in the brain that reflect the template, not the muscles. So let's look for that next. The classic experiment that showed, yes indeed, there are movement objects, used a very clever idea. That idea was to use two different kinds of pliers. Normal pliers, shown on the top half of the screen, is used to pick something up or grab a piece of food when you squeeze the pliers. The bottom of the figure shows reverse pliers. These function exactly the opposite to normal pliers. To open the tongs, you've got to squeeze, and then to grab the object, you relax. The set of muscle movements between the normal and reverse pliers are exactly the opposite to accomplish the same goal. If we could observe a neuron that represented grabbing the food or an object, and it responded the same way using both normal and reverse pliers, then we would have definitive evidence of a template for movement. In other words, a movement object. We see here a monkey using such pliers to obtain bits of food. The top video shows using normal pliers. The red lines coming out of his eyes are a computer animation of where the monkey is looking. This is not Super Monkey. These are not lasers coming out of his eyes, just where he's looking. He's grabbing the food and bringing it to his mouth. Remember, he is squeezing in order to grab the food. Let's take a look at the same monkey using the reverse set of pliers. In this instance, you can see he's relaxing his hand to actually grab the food. In the same monkey, we recorded cells that corresponded to the grabbing of the food. The red arrow shows in time when that was happening. The neuronal activity is shown in two different fashions. On the top part of each graph, there's a little tick mark every time the neuron is active. There's many such trials, so each time the monkey reached for the food, there's a different row of tick marks. Below the tick marks, there's a histogram of activity over all the trials. Let's look at Unit 210. It is very active just as the monkey grabs the food with normal pliers. Likewise, a different neuron, Unit 199, in the same monkey is very active as the monkey grabs the food, again with normal pliers. Now, those same units were observed at the same point in the movement when the monkey was using reverse pliers. Remember, the muscle movements are exactly the opposite. However, the behavior of the unit of the brains are exactly the same. Several other neurons were found that had similar activities. Basically, these neurons code for get the food. They encode for the act of grasping that piece of food, regardless of what muscle movements are involved. And that is an object for movement. Many complex movement objects have been found. They're specific areas where stimulation of the brain gives rise to very specific behaviors. These are movement objects, and they're composed of other objects which ultimately result in the movement of different sets of muscles. There's reaching, climbing, leaping, chewing, defensive postures, moving your hand to your mouth. There's a rich repertoire of behavior here represented as complex movement objects. How does this happen? Well, it's a complex story. Not only are regions of your cortex, the highest parts of your brain, involved, but also non-motor areas involving emotional systems, the cerebellum, systems in your spinal cord, and then finally motor neurons that actually direct the control of contraction of muscles. Many people spend their whole lives studying this system, but I have an analogy that I think will convey to you a good way of thinking about how this whole thing works. Think about a piano. When you push the keys of a piano, there's an amazingly complex set of elements that transform the movement of that key to the sound that you hear. The physics of how this happens is amazingly complex, but the basic idea is straightforward. You push a key and out comes a sound. The keys can be varied and combined with other keys to give rise to new sound objects. I think this gives you a good example or analogy about how movement objects might be represented in your brain. But there's a difference between the machinery in your brain and in a piano. The difference is our brains can learn. Let's look at an experiment. A monkey sits at his desk. He's moving an articulated arm which can have opposing forces applied to it. So it's not a natural motor act from the monkey's point of view. There's a set of numbered diagrams around the tablet. She puts the pen in the middle and then a light, say a number 4, comes on. The monkey has to draw a line from the center out to number 4. But because there's an opposing force, she doesn't do it very well at first. The lines go all over the place. But as the monkey learns, she gets better and better. This is being done unconsciously most of the time. The motor system uses low-level visual feedback as well as proprioception to get better and better at pursuing a goal. It's a form of memory that's different from the way we learn words or phrases or faces, but it's still a form of learning. Such learning is a fundamental property of animal motor systems. Such learning is also being used in the creation of smart prosthetic devices like this one. This amazing robotic hand can manipulate a cube after much learning and practice to show a requested letter. To gain proficiency, it needed decades of experience. Now with a computer, you can do that very quickly. But babies need time to learn movement objects like this, since when you learn a new motor skill, you need to practice. Sometimes the motor part of your brain is smarter than the rest of your brain. Look at the screen. I suspect everybody watching this would say that the orange dot in the middle of the large circles is smaller than the orange dot that's in the middle of the smaller circles. This is a classic illusion called the Ebbinghaus illusion. If you took out a ruler and measured the size of these orange discs, they would measure exactly the same, but they look very different. Now, if I built these discs out of wooden blocks and I put them in front of you and then asked you to reach with your thumb and forefinger and pick up the orange ones, the distance between your thumb and forefinger as you reach for the objects would be exactly the same. So the motor planning part of your brain knows the state of the real world. The visual analysis part of your brain doesn't know the real world. This sets up an interesting dynamic. Different parts of your brain could have different opinions about the state of the real world. How does your brain decide to act? Well, that's the topic of the next seminar. The other item I want to talk about is the difference between moving your muscles and imagining moving your muscles. If you look at the top part of this diagram, it shows the areas of your brain that are active when I ask you to strum your fingers. We would basically see two areas of your brain active. When I ask you to imagine moving your fingers, in other words, don't move your fingers, just imagine you're strumming your fingers, we get the bottom picture of brain activity. Notice one area of the brain is active when you're really moving your fingers as well as when you're imagining moving your fingers. This has significant implications about memory. When you remember moving, you activate some of the same areas of your brain that drive the actual physical movement. The memory activates some of the same areas as the act itself. Not all movements are created equal in your brain. On the left-hand side of this slide, we see areas of your brain that are related to moving parts of your body. There's vastly more of your brain dedicated to your hands, your fingers, your face. That means you have extra capabilities and sensations as it relates to the movements of those muscles and the movement of those limbs. However, this is not the exclusive control of those appendages, as we can see in the diagram on the right. The normal column shows a monkey's grasp of a little piece of food from a well. Fine-grained control of its fingers is present as he grasps the piece of food. If you were to block the monkey's brain from communicating to a spinal cord so that the movement was totally under the control of the spinal cord and not the brain, the monkey wouldn't be completely incapacitated. He would just do so very, very clumsily. So it's not just your brain that coordinates movement, but also your spinal cord. And that's important. Why? Let's take a look. Locomotion motor patterns are reflexes located far away from the upper layers of the brain. They're located in the spinal cord. Here is a rat walking, even after his brain has been chemically separated from his spinal cord. It is the movement of the treadmill that stimulates reflexes in his spinal cord and allows him to walk. These reflexes can be used to offer hope to those who cannot walk due to accidents. We'll talk about this in the last seminar. Fantastic progress is being made using chemical and electrical stimulation of reflexive motor patterns, which offer hope of walking again to those who've lost the ability due to trauma. Isn't that fantastic? Let's review where we are. We began this section noting that people appeared to have a movement template when they wrote this palindrome. It didn't matter what appendage was writing the overall sentence, it clearly was expressing an overall template of movement or a movement object. We then saw the definitive experiment that showed there were neurons in your brain that represented a specific goal of movement, obtaining a bit of food, that was active regardless of which muscles were used to obtain that goal. And then finally, we looked at more complex behaviors that were really movements of movements or objects of objects. Remember that? These represented higher order movements. These movement objects are clearly related to some kind of goal. Grasping an object, defending against a threat, bringing something to the mouth, chewing a bit of food. So yes, there clearly are movement objects. We can use our Homer analogy to explain this in a simple statement. You plan with movement objects, you move with muscle stuff. Almost the exact mirror image of our sensory analysis. We said that you sense stuff and perceive objects. Your brain's sensory systems go from low-level analysis to higher-order objects. The movement system is the mirror opposite. Your brain is planning acts based on high-level movement objects, and they get translated by this very complex piano-like machinery into muscle stuff. Some muscles contract, some muscles expand, there's feedback, there's pattern generators for complex behaviors like locomotion, etc. The essential fact here is that your brain is planning behavior with movement objects. The objects get translated to muscle stuff depending on your position in space. Isn't that cool? That completes our first seminar. The topic we'll talk about next is illustrated by this quote from Amelia Earhart, a very famous American pilot. She said, "The most difficult thing is the decision to act. The rest is merely tenacity." We saw today how the brain can have different opinions. Is the size of that orange disc the same or different? How does the brain come to a decision about how to act about that? Today, we studied the movement system, but the decision to act had already been made. Our next seminar is going to be the decision-making processes of your brain as it decides to act. I hope you enjoy the seminar. See you next time. This is Steve Saltwick.